The I-SPY2 trial is an adaptively randomized phase 2 trial with the goal of finding effective drugs to take into phase 3 in the breast cancer setting. We use neoadjuvant therapy, which is therapy that we can use before surgery, in an effort to see if we can improve the rate of pathologic complete response. This was a combination of drugs for patients with HER2 positive breast cancer. So we looked at the combination of T-DM1 and pertuzumab and compared that to the standard therapy with paclitaxel and Herceptin. All patients then went on to get the standard adriamycin and Cytoxan and then had surgery. What we found in the trial was that the combination of T-DM1 and pertuzumab had a dramatically increased effect on bringing patients to the pathologic complete response endpoint. We had about a 30% increase in the PCR rate, and we have a very unique endpoint in the I-SPY2 trial. It's not simply to find the rates at which we would develop pathologic complete response, but because it's a Bayesianly designed, adaptively randomized trial, we have the ability to generate predictive probabilities. And the endpoint is to have a predictive probability greater than 85% that if this drug works in I-SPY2, it will subsequently work in a randomized phase 3 trial. And so our endpoint is intimately linked with the goal of finding effective therapies and taking them to phase 3 so that they can be found, we can determine whether or not they're going to benefit patients. Well, I think that what we aim to do is to try to speed the process of finding effective drugs, but also speed the process of finding ineffective drugs. And that's where, again, the adaptive design is very useful, because we're constantly evaluating how each patient in the trial is doing and how each drug is performing over time. Those drugs that are doing well, performing well, will have more patients randomized to their arms, and they will accumulate faster, and we will get those answers faster. And the great thing about this, having a surrogate like pathologic complete response, means we have a short-term endpoint that fairly well predicts a long-term endpoint. That is reduction in recurrence and overall survival. And that's something that the FDA is really looking for, is ways in which we can find the winners, we can use an early surrogate, which can enable them to give accelerated approval to a drug and get it to patients when it looks promising, while at the same time going through the confirmatory trial process in phase 3. It's a standing platform trial, which means we can be evaluating multiple drugs at the same time from multiple companies, which is very unusual. But because we have a standard platform, because we have a master IND, because we essentially have an honest broker, different companies are willing to come into the trial to test their drugs. And so that has enabled numerous drugs to come through this mechanism over the last 6 years simultaneously. And this is the approach that was taken for pertuzumab. It was found to have a dramatic impact on pathologic complete response. The FDA gave it accelerated approval. Patients are now having access to that drug while we wait for the long-term outcome studies that will come later. And so the hope is that by finding the winners early and quickly, we can do a better job of bringing effective drugs to patients in a shorter time span, in a less expensive way, and ultimately in a way that's going to help us start to build upon these backbones and bring less toxic drugs to this effort. And that's the other big news from this particular arm of the trial, is that not only did the combination of T-DM1 and pertuzumab increase pathologic complete response rates by about 30%, but it did so with less toxicity to the patients. Because we were able to remove the paclitaxel from the standard regimen in this investigational arm, we found that patients did not have as much in the way of alopecia or neuropathy, two very, very bothersome side effects that not only affect them while they're getting the drugs, but also can be long-term effects. Neuropathy can be debilitating for women for decades. So we really want to try to find drugs that are particularly targeted to the unique biology of these tumors and that aren't going to have toxic effects on all of the normal cells in the body. And that was a really great additional finding to this arm of the I-SPY2 trial, to find that T-DM1 pertuzumab was not just more effective in reaching PCR than standard of care, but actually did it with less toxicity, particularly the kinds of toxicities that really matter to patients. Certainly. So in I-SPY2, the overall trial, we're not limited to just HER2 positive patients. We test drugs in basically every subset. And I think this has been a very useful exercise to help others in other diseases think about how can they set up a standing trial when we now start to understand the molecular phenotypes within a disease and say, okay, now we're going to have this trial going. We're going to test different drugs against different targets simultaneously. And the MATCH trial is a great example of a trial that's doing just that, not just in breast cancer, but in a variety of diseases. I think this is a really exciting time because now we have the tools to be able to bring drugs to patients in an earlier part of the disease trajectory. We used to develop drugs by giving them to patients who had stage 4 disease, who'd already gotten a lot of other therapy. And now we understand that the biology of metastasis is very different than biology of a newly diagnosed tumor. And so now we can test drugs in those patients with newly diagnosed tumors, and we may find that drugs work that would never have worked in the metastatic setting. It's really apples and oranges. And so focusing our drug development efforts in patients who can really benefit from it, really almost to improve cure rates for certain diseases, is really exciting. And I think we're going to see more and more of this as we have more targeted therapies in different diseases.